Imagine looking into just a stainless steel vat of cheap, cloudy liquid algae.
Right, just kind of a green sludge.
Exactly, just green sludge. But then someone shines a highly complex three-dimensional hologram of blue light directly into that water. And
Okay.
right before your eyes, a perfectly structured, self-repairing computer processor literally crystallizes out of nowhere.
Wow.
No billion-dollar fabrication plants, no toxic silicon mining, just light, water, and biology.
It sounds like pure magic, honestly. Or maybe, science fiction from a century in the future, but the mechanics behind that image are actually rooted in science that is happening right now.
And that is exactly why we are here today. Welcome to this deep dive. Today, we're taking you on a journey that is going to completely dismantle and then rebuild your understanding of the physical world.
Yeah, it's a big one.
It really is, because usually when we talk about biology, whether it's a disease or a forest or a virus, we treat it as this messy, unpredictable force of nature. It's something we just have to endure or suppress or fight.
Right, we use the language of war constantly. I mean, we talk about battling cancer, eradicating a virus, or conquering an invasive species.
Yeah, exactly.
We view biology as an adversary that lacks precision. We prefer the clean lines of human engineering, concrete, steel, binary code.
But what if that entire worldview is just backward? What if everything we think we know about medicine, manufacturing, and environmentalism is based on a fundamental misunderstanding of what biology actually is?
That's the core question here.
Right. What if biology isn't a messy enemy at all, but rather the most advanced, programmable technology in the universe?
And to explore this, we are diving into a stack of materials centered around a really fascinating series of speculative science essays. They were written in 2026 by an independent researcher named Zachary David Enfield.
When I first looked at the source material, I honestly thought our researchers had accidentally mixed up three completely different deep dives.
It's a lot of ground to cover.
It really is. Because over here, you have essays on molecular biology and gene therapy. In the middle, there's a deep dive into material science and quantum electronics.
Yeah.
And then on the other end, there's stuff about agricultural ecology and wastewater management.
Yeah, it looks completely disjointed at first glance. But that is the mission of our deep dive today. We are going to connect the dots across all of these incredibly diverse fields.
Which is wild.
It is. Our goal is to show you how reprogramming human cells to cure disease, rebuilding our industrial materials from scratch, and rescuing our collapsing planetary infrastructure are all, underneath it all, the exact same engineering problem.
I promise you, by the time we wrap up today, you will look at a virus, a diseased leaf, or a polluted pond in a completely different way. Not as a threat, not as a tragedy, but as an opportunity for reprogramming.
Right. But to get to that macroscopic level, we have to start at the absolute foundation. Before we can reprogram the world, we have to understand the code running inside our own bodies.
Makes sense.
We have to shift from this grand philosophical premise to the microscopic reality of human disease.
So let's talk about disease, because Enfield starts by arguing that conditions like cancer, autoimmune disorders, and prion diseases aren't these monsters growing inside us.
Right, they're not alien invaders.
They're simply failures of information. They're corrupted code.
It's a really profound reframing, and we need to look at it mechanistically to really grasp it. Let's take cancer.
Okay.
A cancer is not an invader. It is a population of your own cells running a corrupted version of the genome's growth regulation program.
So it's our own hardware running bad software.
Exactly. A healthy cell operates on a very tight regulatory loop. It grows, it divides, it pauses to check its own DNA for errors, and if it finds too many errors, it undergoes a process called apoptosis.
I've heard that term, apoptosis. It's programmed cell death. Right, like the cell realizes it's broken and essentially falls on its sword for the good of the body.
That's a great way to put it, yeah. It's a suicide protocol written into the DNA, governed by specific genes like p53, which acts as sort of the guardian of the genome.
Oh, interesting.
But in cancer, that code is mutated, the loop is broken, the growth signals get stuck in the on position, and the cell cycle kill switches like p53 are completely silenced or deleted.
Okay, let's unpack this with a software analogy. It's like a glitch in a computer program that forces an application to instantly copy itself in the background, right? Until it uses up all the RAM and crashes the entire hard drive.
That is exactly what it is. And Enfield applies this information theory lens to other diseases, too. Take autoimmune disorders like rheumatoid arthritis or lupus.
What's his take on those?
He calls that misclassification logic. Your immune system has an incredibly complex database of what is self and what is foreign.
Right.
In an autoimmune disease, the code responsible for tagging tissue misfires, and it accidentally tags your own joint cartilage as a foreign pathogen.
Wow. And prion diseases. Things like mad cow. Because those always sounded terrifying.
Oh, they are. But mechanistically, they are structural errors propagating like a corrupted data type.
How does that actually work?
Well, a single protein folds into the wrong three-dimensional shape. And when it bumps into a correctly folded protein, it forces that healthy protein to adopt the broken shape. It's a cascading physical glitch.
Okay, so if we accept this premise that disease is fundamentally an information problem, it completely changes the solution.
That really does.
Because if cancer is a corrupted software loop, the best intervention shouldn't be just hacking it out with a surgical scalpel or poisoning the whole body with chemotherapy. The intervention should be debugging. You need to patch the code.
Yes. But to patch the code, you run into the greatest physical barrier in medicine. How do you deliver a new set of instructions past the cell membrane, through the cytoplasm, and securely into the nucleus of a living human cell?
Right. Because our cells are heavily fortified. They don't just let random pieces of DNA float in.
Exactly. They actively destroy foreign DNA. It's how we survive. So, human engineers have spent decades trying to build microscopic delivery vehicles, lipid nanoparticles, synthetic polymers, just to sneak the code in.
And we are making progress on that, right?
We are. But Enfield points out something entirely obvious in hindsight. We don't need to invent the ultimate delivery vehicle. It already exists.
And this is where the text completely flips the script on viruses.
Yeah.
I mean, we've spent our whole lives, especially recently, terrified of viruses.
Yes.
We view them as the ultimate biological villains. But Enfield strips them down to their purely mechanical essence. A virus is just a molecular courier.
Let's look at the physical structure of a virus. Strip away the pathology for a second. A virus consists of a geometric protein shell, the capsid surrounding a strand of genetic material.
That's it.
That's it. It has no metabolism, it doesn't eat, it doesn't move on its own. Its entire evolutionary purpose is to bump into a specific host cell, trick the cell's receptors into opening the door, and inject that genetic material.
Okay, think of a virus like a hijacked FedEx truck.
I love that analogy.
The truck itself is an absolute engineering marvel. It knows exactly how to navigate the microscopic highways of the bloodstream. It knows how to bypass the immune system's security checkpoints.
Right.
It knows how to dock perfectly at the target destination, say a liver cell instead of a lung cell. And for years, medicine has basically been trying to blow up the trucks.
Because the payload inside the truck has always more viruses until it bursts.
But now we're realizing we don't have to blow up the trucks. We just open the back, take out the explosive package, and swap in a repair manual.
Exactly. And when you do that, you inherit 3 billion years of evolutionary optimization for free. The virus has already solved the hardest problems in nanoscale drug delivery.
Like what?
Well, it knows how to cross the lipid bilayer. It knows how to avoid being dissolved by cellular enzymes. When you strip a virus of its harmful genes and load it with therapeutic code, it ceases to be a pathogen. It becomes a nanite, a biological machine built for healing.
And if you're listening to this thinking, wow, that sounds like a great concept for a sci-fi movie in 50 years, you need to understand that this is clinical reality right now.
It really is.
The source material lays out the data. By 2024, there were already 7 FDA-approved gene therapies using what are called adeno-associated viruses, or AAVs.
Right. AAVs are fascinating. They're small viruses that naturally infect humans, but they are generally non-pathogenic. They don't cause disease on their own. That makes them the perfect chassis for our FedEx trucks.
Let's talk about what these modified AAVs are actually doing in patients. Take Luxturna, which was approved back in 2017. This completely blew my mind.
It's incredible.
It cures a specific type of inherited blindness. How does it actually work?
Well, the patients with this condition have a mutation in a gene called RPE65. This gene is responsible for producing an enzyme in the retinal pigment epithelium, which is a layer of cells at the back of the eye.
Okay.
This enzyme is crucial for the visual cycle. Without it, the photoreceptors in the eye essentially starve, and the patient slowly goes blind.
So it's a missing line of code.
Exactly. So scientists take an AAV, gut its viral replication code, and insert a pristine, functional copy of the RPE65 gene. The surgeon injects these viral nanites directly beneath the patient's retina.
Oh, wow. So right into the eye.
Right. The viruses recognize the retinal cells, bind to them, get pulled inside, travel to the nucleus, and deposit the functional gene. The cell reads the new code, starts manufacturing the missing enzyme, and within days, the patient's vision is restored. A single injection.
That is miraculous. And it's not just the eyes, right? There's Zolgensma, which targets spinal muscular atrophy in infants.
Yes.
This used to be a uniformly fatal neurological disease where motor neurons die because of a defective SMN1 gene. Babies would slowly lose the ability to move, swallow, and eventually breathe.
It was devastating.
But Zolgensma uses an AAV to deliver a working copy of that gene across the blood-brain barrier. It completely halts the deterioration.
It does. But as incredible as gene replacement is, dropping off a working copy of a gene, the real paradigm shift happens when you combine these viral delivery trucks with CRISPR.
Right.
Because CRISPR allows you to not just add a new file, but to actively edit the existing code.
I feel like everyone has heard the word CRISPR in the news, but the text really gets into the mechanics of it, and we have to talk about the history here, because it's the perfect example of how weird and amazing biology is.
Oh, the history is wild.
CRISPR wasn't invented in a shiny biotech lab by a supercomputer. It was discovered in yogurt, right?
Yes, specifically in the bacteria Streptococcus thermophilus, which is used to make yogurt and cheese. The dairy industry was having a massive problem with bacteriophages, viruses that infect and kill bacteria, basically wiping out their bacterial cultures.
Okay.
And researchers studying these bacteria noticed they had this bizarre immune system.
A bacterial immune system, which sounds like an oxymoron.
It does, but bacteria have to fight off viruses just like we do. When a virus attacks a bacterium and injects its DNA, if the bacterium survives, it actually snips out a tiny piece of that viral DNA and saves it in its own genome in a clustered sequence.
Like keeping a record of it.
Exactly.
Yep.
That sequence is the CRISPR array. It's essentially a molecular mugshot database.
Wanted. Dead or alive.
Precisely. If that same virus ever attacks again, the bacterium transcribes that mugshot into a guide RNA. This guide RNA acts like a bloodhound. It pairs up with an executioner protein called Cas9.
Okay, I love these names.
Right. So the guide RNA sniffs around the cell, and if it finds a piece of DNA that perfectly matches the mugshot, the Cas9 protein locks on and acts like a pair of molecular scissors. It physically cuts the viral DNA double helix in half, completely neutralizing the threat.
So human scientists looked at this microscopic bacterial defense mechanism and realized, wait, if we just swap out the mugshot, if we give it a guide RNA that matches a human genetic mutation instead of a virus, we can send these molecular scissors to any specific address in the human genome.
And that is exactly what we did. We repurposed an ancient bacterial immune system into a programmable text editor for DNA.
Incredible.
When Cas9 makes a targeted cut at the site of a mutation, the human cell panics. It sees a broken DNA strand and rushes to repair it. If we simultaneously flood the area with the correct template of that gene, the cell will use our template to stitch the DNA back together. You have permanently patched the code.
The text highlights the absolute watershed moment for this, which is the FDA approval of Casgevy in December 2023. This was the first CRISPR therapy approved for humans, and it cured sickle cell disease.
And the mechanism of Casgevy is brilliant because it's actually a workaround. Sickle cell disease is caused by a single letter typo in the gene that makes adult hemoglobin.
Just one letter.
Just one typo. But it causes the hemoglobin proteins to stick together and form rigid chains, deforming the red blood cells into a sickle shape that clogs up blood vessels and causes agonizing pain.
So do they use CRISPR to fix that one typo?
Actually, no. They use CRISPR for a much more elegant hack.
Okay.
We all have a second hemoglobin gene, fetal hemoglobin, which we use in the womb, but it gets turned off shortly after birth by a genetic switch called the BCL11A gene. Casgevy uses CRISPR to cut the BCL11A gene, permanently breaking the off switch. The body just reverts to producing fetal hemoglobin, which doesn't sickle.
It's like finding a corrupted file in an operating system,
Right.
realizing you have a perfect backup from an older version of the OS, and just redirecting the system to boot from the backup.
Exactly.
And the results are staggering. Out of 31 patients in the core trial, 29 were completely free of severe pain crises for over a year following a single treatment.
And the trajectory of this technology is moving incredibly fast. Casgevy is an ex vivo treatment. They take the patient's bone marrow cells out, edit them in a lab, and put them back in. But the sources highlight the next wave, which are in vivo treatments. That's where the editing happens directly inside the patient's body.
The FedEx trucks driving right through the bloodstream.
Right. There's NTLA 2001, which uses lipid nanoparticles to deliver CRISPR directly to the liver to knock out a gene causing hereditary amyloidosis.
What does that disease do?
It's a fatal disease where misfolded proteins build up in the heart and nerves. But with this treatment, they achieved a 90% reduction in the pathogenic protein.
90%. Wow.
Then there's CTX310, another in vivo CRISPR therapy that permanently switched off the ANGTTL3 gene in the liver, dropping patients' cholesterol by 50% and triglycerides by 55% in clinical trials.
Imagine never taking a statin pill again because you
Yeah.
just permanently edited your liver's cholesterol production code.
Yeah.
It's wild to think about.
It is.
But let's take this back to cancer. Because Enfield proposes a concept for a cancer treatment that sounds like military strategy mixed with IT support. He calls it the three-mode viral nanite architecture.
This is a brilliantly integrated platform. It shows what happens when you fully master biological programming. The first mode is seek and destroy.
How does that work?
You biopsy a patient's tumor, sequence its genome, and identify the unique mutations driving it. You then program your viral nanite with guide RNAs targeting those specific mutations. The nanite circulates through the body, completely ignoring healthy cells. But when it enters a cancerous cell and identifies that mutated signature, it delivers a genetic kill switch, triggering that apoptosis pathway we talked about earlier.
So it's a smart bomb that only detonates if it reads the exact barcode of the tumor. I mean, that alone would cure cancer. But Enfield doesn't stop there. Then comes the restore mode.
Yes. The same delivery platform can be programmed to release localized signaling molecules, like specific cytokines, that activate regeneration pathways in the surrounding healthy tissue.
So it cleans up the mess.
Right. It essentially coaxes the local stem cell populations to wake up, divide, and replace the structural void left by the cancer cells that were just destroyed.
And finally, there's the self-destruct mode. Because obviously, if you have millions of programmed viral nanites floating around your body, you don't want them lingering forever.
Precisely. The nanite's genetic payload is built with synthetic promoter sequences. Think of a promoter like a mechanical counter.
Okay.
Every time the cell divides or every time the nanite activates, the counter ticks down. After a certain number of cycles, the promoter permanently deactivates transcription. The viral machinery shuts itself off and is quietly recycled by the body.
I want to pause here and make sure we really absorb the magnitude of this.
Yeah.
Why is this shift from chemical medicine to information medicine so important?
It really comes down to the medium we are working with. DNA is the most absurdly dense information storage system ever documented in the universe. A single gram of DNA can hold over 200 petabytes of data.
I can't even visualize a petabyte.
To put it in perspective, 200 petabytes is roughly equivalent to the entire archive of digital photographs ever taken by humanity, all stored in a drop of liquid roughly the size of a Skittle.
Oh my God.
Right. When you realize that the human body is essentially a sprawling network of hyperdense hard drives communicating through chemical logic gates, you realize how crude traditional medicine is. We have been trying to fix corrupted software by pouring various chemicals on the motherboard.
Throwing a wrench at the computer and hoping it unsticks the keyboard.
Exactly. We're moving from blunt chemical warfare against the body to the precision patching of corrupted code.
It completely reframes our relationship with our own biology. But here is the massive pivot in the source material. If we have these viral couriers and we have CRISPR, well, if we can write a patch to fix a broken line of code, what happens when we use that exact same delivery system to install brand new software? We shift from healing a disease to deliberate human augmentation.
This is where we cross a major conceptual threshold. And it's vital to understand that from an engineering perspective, the delivery system does not care.
It's just a truck.
Right. The viral vector doesn't know if it's carrying a gene to fix a sickle cell mutation or a gene to give you a completely new biological capability. The physical mechanics are identical. The difference is solely in the intent and the payload.
And Enfield argues that we don't even need to be super geniuses inventing these new capabilities from scratch because nature is essentially a massive open source repository of code.
3 billion years of beta testing.
Exactly. Evolution has spent all that time running trillions of parallel experiments, solving problems that humans struggle with: extreme radiation, freezing temperatures, lack of oxygen.
This brings us to a concept Enfield calls functional translation. In the early days of genetic engineering, if you wanted a trait from a jellyfish, you might literally cut and paste the jellyfish gene into a mouse and cross your fingers, hoping the mouse's cellular machinery could read it.
Like those classic glowing green mice.
Right. But synthetic biology does something much more elegant now. It reads the biological solution from an organism, understands the chemical mechanism of how that protein works, and then synthetically writes a completely human-optimized version of that gene sequence.
Wait, really? So you don't even need the original animal DNA?
No, you don't transplant the animal's code. You borrow the meaning of the solution, and you write the implementation bespoke for human cells using the codon sequences that human ribosomes prefer. The species barrier just completely dissolves.
The examples he gives from the trait library are just mind-blowing. Let's start with tardigrades, the famous water bears.
Oh, they're amazing.
These microscopic creatures can survive being boiled, frozen, dried out for decades, and exposed to the vacuum of space. But the crazy part is they can survive radiation doses 2,000 to 3,000 times higher than the human lethal limit.
And researchers found the code for how they do it. It's a protein called DSUP, which stands for damage suppressor.
How does it actually work? Does it act like a microscopic lead shield?
Sort of, but it's more dynamic than that. DSUP is what's called an intrinsically disordered protein. It doesn't have a fixed, rigid 3D structure. Instead, it acts like a flexible protective cloud.
A cloud.
Yeah, it binds directly to the nucleosomes, which are the spools that DNA wraps around, and physically hugs the DNA double helix. When high-energy radiation hits a cell, it usually creates reactive oxygen species, free radicals that act like molecular shrapnel, shredding DNA.
Yikes.
The DSUP protein essentially absorbs that shrapnel. It neutralizes the free radicals before they can break the DNA strands.
And this isn't just theoretical biology, is it? The sources detail how human cells engineered in a petri dish to express this DSUP protein showed 40% more tolerance to severe X-ray radiation.
That's right.
But what really got me was the MIT research from 2025. They demonstrated that you don't even need to permanently edit a person's genome to give them this superpower.
Yes, the temporary augmentation approach. They delivered synthetic mRNA encoding the DSUP protein using lipid nanoparticles.
Just so we're all on the same page, lipid nanoparticles, LNPs, are the exact same technology used in the mRNA COVID vaccines, right?
Yes, exactly. An LNP is basically a microscopic bubble of fat. Because our cell membranes are also made of lipids, when the LNP bumps into a cell, the two fat layers merge seamlessly, and the mRNA payload is dumped inside.
Oh, that makes so much sense.
The cell reads the mRNA, manufactures the DSUP protein for a few hours, protects the DNA, and then the mRNA naturally degrades. There is no permanent change to the genome.
So you could theoretically give a patient an injection to temporarily armor their healthy tissue right before blasting a tumor with radiation therapy.
Exactly.
Or you could give an astronaut a localized shot to protect their bone marrow right before a solar flare hits their spacecraft. It's augmentation on demand. What about enhanced vision?
Well, we know that owls have specific opsins, which are light-sensitive retinal proteins that are highly tuned for low-light environments. Insects and certain snakes have receptors that allow them to see into the ultraviolet and infrared spectrums.
Wait, are you seriously saying we could inject a virus into someone's eye, they'd suddenly see in infrared? That sounds terrifying. Wouldn't the human brain just reject that input?
It's a valid concern, but neuroplasticity is incredibly robust. We know from experiments with colorblind monkeys that if you introduce a missing color opsin gene via an AAV vector, the brain's visual cortex eventually learns how to process the new signal.
It just adapts.
Right. Because we already have the AAV delivery vectors perfectly validated for retinal delivery, remember Luxturna, we could theoretically deliver synthetic, human-optimized versions of infrared opsin genes directly to human photoreceptors.
That is wild.
The rods and cones would produce the new protein, and when an infrared photon hits it, it would fire an action potential down the optic nerve. Your brain would just have to learn to map that new color.
Okay, what about oxygen efficiency? Elite freedivers train for decades to maximize their oxygen use, but a Weddell seal can just casually hold its breath for over an hour.
They have evolutionarily hacked oxygen storage. Mammals use a protein called myoglobin to store oxygen in muscle tissue. Humans have a baseline level of it. Marine mammals have up to 30 times the myoglobin concentration in their muscles.
And we know the genetic architecture for that?
Fully mapped. It's largely a matter of regulatory elements, the code that tells the body how much of the protein to make. If a viral nanite delivered those regulatory sequences to human muscle tissue, upregulating myoglobin production, the implications would be revolutionary.
I imagine not just for deep-sea diving, right?
Right, for high-altitude mountaineering or for patients with severe pulmonary diseases like COPD who are constantly starved for oxygen.
But the trait that really messes with my head is regeneration. If a salamander loses a leg, it doesn't just form a scump. It forms this mass of undifferentiated cells called a blastema and just regrows the whole limb. Bone, muscle, blood vessels, nerves, all perfectly patterned and seamlessly integrated. I always assumed humans just fundamentally lack the biological hardware for that.
That has been the prevailing assumption in medicine for a century. But recent research turns it completely upside down. A 2025 study traced the initiation of salamander limb regeneration to the sympathetic nervous system's adrenaline signaling pathway. It's the exact same adrenaline pathway that humans have.
So why don't we regrow our arms?
Evolutionary trade-offs. Mammalian evolution prioritized rapid wound sealing. If an early mammal got its arm bitten off, the risk of bleeding to death or getting a lethal infection was massive. So our bodies evolved to rapidly deploy fibroblasts to create scar tissue and seal the wound instantly.
Oh, I see.
And scar tissue is the enemy of regeneration. It physically blocks the blastema from forming.
So the body basically says, I don't have time to carefully rebuild a hand. I just need to plug this hole right now so we don't die.
Exactly. But research out of LSU suggests that humans still possess the entire underlying genetic toolkit for limb regeneration. The code for how to build an arm is still in every single cell of your body. That's how you grew arms in the room in the first place. The regulatory elements that would reactivate those pathways in an adult have just been suppressed. They are locked away.
It's just dormant code.
Yes. A targeted viral nanite could potentially deliver specific transcription factors, which are proteins that turn genes on or off to suppress the scar formation pathway and simultaneously unlock the dormant blastema formation code.
So we are talking about moving from natural evolution to programmable evolution.
That is the crucial distinction at the heart of Enfield's work. Natural evolution is undeniably powerful, but it is blind, gradual, and incredibly wasteful. It relies on random mutations over millions of years, most of which are fatal. Programmable evolution replaces that random, bloody walk with directed, intentional navigation.
That makes sense.
Enfield outlines a modular platform for biological versioning to make this a systematic engineering discipline. It has 5 layers.
Let me walk through these because the structure really grounds this crazy science. Layer 1 is the trait library. We catalog genetic modules from across the tree of life like DSUP and myoglobin and synthetically rewrite them for human ribosomes. Layer 2 is delivery engineering, perfecting the viral vectors to target very specific tissues like AAVs that can cross the blood-brain barrier without triggering an immune response.
Pivotal step.
Layer 3 is the editor, CRISPR and its next-generation successors that make the actual microscopic DNA cuts and inserts. Layer 4 is dynamic control. This is huge. Building synthetic switches so a gene only turns on when a specific condition is met, like the presence of a tumor marker or a spike in radiation.
Exactly.
And layer 5 is iterative augmentation. Doing this gradually, monitoring the human immune response, adding features one by one, rather than trying to rewrite the whole genome at once.
It's a comprehensive engineering framework. It gives humanity a model of the future and the intention to build it, which natural selection entirely lacks.
Okay, I have to step in here and throw a massive red flag on the play. This is based directly on Enfield's own warnings in the text.
Let's hear it.
If we start authoring our own biology, are we risking a permanent biological class divide? You mentioned earlier that Casgevy, the sickle cell cure, costs $2.2 million a dose. If these augmentations cost millions of dollars, won't this just create a dystopian society of genetically enhanced billionaires and unenhanced, fragile, regular people? We're talking about taking economic inequality and literally writing it into human DNA.
It is the most critical ethical question of the century, and Enfield does not shy away from it. He insists that safeguards must be built in as hard architectural requirements, not as regulatory afterthoughts that we tack on later.
How do you architect an ethical safeguard into a virus?
First, technical safeguards. Every viral nanite must carry a verified kill switch, allowing the process to be halted instantly by a specific inert chemical trigger. Second, reversibility is key.
Like the temporary mRNA stuff?
Yes, this is why transient mRNA enhancements, like the radiation shield that fades over a few hours, carry a completely different ethical weight than permanent genomic changes. If you can opt out, the coercion factor drops.
Right, but what about the permanence of editing the human race?
This is the core ethical architecture, the strict hard line between somatic edits and germline edits.
Define the difference for us.
Somatic edits change the cells in an individual's body, like fixing the liver or the retina. Those changes are localized to the patient. They affect only the consenting adult, and they die when the adult dies. Germline edits, on the other hand, change the DNA in embryos, sperm, or eggs.
So they get passed down.
Yes. That means the edits are inherited by all future descendants. You are making choices for people who do not yet exist and cannot consent. Enfield argues for a strict, heavily enforced international moratorium on all germline augmentation until global governance frameworks mature.
And what about the cost? The $2.2 million price tag.
He argues that enhancement access must be treated as a public health question, not a luxury market. But the economics of biology are unique. The clinical infrastructure being built right now to cure terrible diseases, the bioreactors, the delivery vectors, the clinical trials, will inevitably crash the cost curve.
So the early stuff subsidizes the future stuff.
Medicine and enhancement aren't competing for resources. Medicine is building the paved highway that augmentation will eventually drive on.
Okay, so we've spent all this time talking about reprogramming the software of life inside our bodies. But Enfield pivots here. He argues that to build a new civilization, we don't just need better bodies. We need new hardware. We need new physical materials to build our world out of. And ironically, the ultimate building block for the future turns out to be the exact same biological mechanism behind some of our most terrifying diseases.
We are moving from the software down into the physical substrate. To understand this, we need to look closely at prions and amyloids.
If you're listening to this and picturing mad cow disease right now, you aren't alone. I had the exact same thought. Prions are the misfolded proteins that eat holes in your brain. Are we really talking about building airplanes and computers out of prions?
It sounds absurd, but Enfield asks us to separate the mechanism of a prion from the disease it happens to cause in mammals. Prions and functional amyloids share a fundamental structural motif known as the cross-beta architecture.
Let's break that down. What does cross-beta architecture actually look like under a microscope?
Imagine a slinky, but instead of coils, it's folded back and forth like an accordion into flat sheets. These are called beta strands. In a prion fiber, these beta strands stack on top of each other, oriented perpendicular to the long axis of the fiber.
Okay, I can picture that.
Crucially, they are locked together by an incredibly dense network of hydrogen bonds.
Hydrogen bonds are the same forces that hold water molecules together, right?
Yes, but here they are packed so densely they form an almost impenetrable lattice. And sandwiched between these rigid hydrogen-bonded sheets are the protein's side chains, which are the floppy chemical arms hanging off the amino acids. These side chains interlock like a zipper, acting as microscopic shock absorbers.
So you have alternating layers of ultra-rigid hydrogen bonds separated by flexible shock absorbers. It sounds like a nanocomposite material.
It is the exact same design logic you see in an abalone shell or mother of pearl, which are incredibly tough but operating at the 10-nanometer scale. And the mechanical consequences of this geometry are extraordinary.
Let's look at the numbers because they are genuinely insane. The text notes that engineered polymeric amyloid fibers have a toughness of 160 megajoules per cubic meter. For context, that beats spider dragline silk, which has always been the holy grail of biological materials, and it achieves this at 1/8 the density of steel.
And the beauty of this material is that human engineers have a simple, programmable design dial to control its properties. It's called hydrogen bond density, or HBD.
Think of HBD like the thread count in a luxury hotel sheet. If you weave the threads really loosely, you get a light, breathable linen. If you pack them incredibly tight, you get something like ballistic Kevlar.
That's a perfect analogy. If you engineer the amino acid sequence to pack the residues as tightly as physically possible, you get high HBD. That gives you a structural steel analog. It is so stable that these engineered amyloid fibers can survive boiling in concentrated acid, exposure to pure formic acid, and extreme gamma radiation that would instantly fry a silicon microchip.
But if you turn the dial the other way,
low HBD gives you a more dynamic, fragile fibril. This is incredibly useful for things like dynamic biosensors inside the body that need to assemble, to take a chemical, and then rapidly disassemble so they don't clog up a blood vessel. You have one single self-assembling biological system that can be tuned across a vast mechanical spectrum.
Here's where it gets really interesting. We're talking about a biological material that conducts electricity, generates power from sound, and physically grows itself. How does this change the future of computers? Because silicon feels very clunky compared to this.
It changes everything about hardware. Let's look at the electrical properties first. Because these fibers form perfectly straight 10-nanometer wide tubes, you can metallize them. You use the prion fiber as a self-assembling scaffold to grow a perfect gold or silver nanowire.
But the text says you don't even need the metal. The protein itself can conduct electricity. How does a piece of protein act like a copper wire?
through a quantum mechanical process called tyrosine hole hopping.
Okay, stop right there. You cannot just drop tyrosine hole hopping into the conversation without a breakdown. What is a hole in electron transport, and what is tyrosine doing?
Fair point. Tyrosine is an amino acid that has a ring-shaped chemical structure called an aromatic ring. In these densely packed prion fibers, the tyrosine rings stack up directly on top of each other, incredibly close together, like a stack of coins.
All right, stack of coins, got it.
Now, when we talk about electricity, we usually think of an electron moving down a wire. But quantum mechanics allows for the opposite. Imagine a bucket brigade of people passing a bucket of water. Instead of looking at the bucket moving forward, imagine looking at the empty space left behind by the bucket. That empty space, the hole, moves backward down the line. In these stacked tyrosine rings, an electron gets knocked out, creating a positively charged hole. That hole can jump or hop from one ring to the next across micrometers of space.
Oh, I see.
It allows the protein fiber to intrinsically conduct an electrical current without any metal at all.
That is mind-bending. And they can also conduct protons.
Yes, which makes them the perfect membranes for biocompatible fuel cells that run entirely on biological fluids.
And then there's the piezoelectric effect. Piezoelectricity is when a material generates an electrical charge when you physically squeeze it or apply mechanical stress to it.
Right, it's how quartz watches work or how the igniter on a gas grill sparks. These engineered amyloids are highly piezoelectric. They can generate up to 1 volt of open circuit voltage just from being hit with ultrasound waves.
The text mentions a medical application for this that sounds like witchcraft. You can inject these nanostructures into a tumor and then just hit the tumor from the outside with standard ultrasound, like what they use to look at babies in the womb.
Yeah.
The sound waves squeeze the amyloids, they generate a localized electric field, and that electricity breaks water molecules apart to create reactive oxygen species, which literally zap the cancer cells to death. No batteries, no wires, no incisions.
It's purely exploiting the physical geometry of the protein. And because these are proteins, you can manufacture them at essentially zero marginal cost using a process called seeding.
Right, because you don't build these fibers in a multi-billion-dollar semiconductor fab with workers in bunny suits and toxic hydrofluoric acid.
No, you take a massive bioreactor, fill it with a cheap liquid protein feedstock, which we can harvest from algae, and you drop a tiny microscopic seed of the preformed fiber into the vat. You agitate the liquid. The seed bumps into the liquid protein monomers, forces them to adopt the cross-beta shape, and the fiber grows exponentially. It just crystallizes out of the liquid.
So instead of a factory, you have a brewery. You just brew structural material.
Precisely. And macroscopic applications follow naturally. Imagine self-repairing aerospace hulls. If a micro crack forms in an amyloid composite panel of an airplane, the broken ends of the fibers are exposed to the surrounding protein matrix. The seeding cascade instantly restarts automatically, growing new fibers to knit the crack closed in midair. We already see this exact mechanism in bacterial biofilms in nature.
Let me ask the obvious safety question again, just to be absolutely sure. Are we building aerospace hulls out of mad cow disease? If I scrape my hand on the airplane wing, do I get a lethal neurological disease?
Enfield addresses this containment issue directly, and the resolution is clean. Engineers do not use mammalian prion proteins. They use harmless yeast prion determinants like the SUP35 protein or completely synthetic, artificially designed peptides that have zero biological activity in the human body.
So it's safe.
It's perfectly safe. The engineering borrows the architectural mechanism of self-assembly, but it leaves the pathology completely behind.
Okay, so going back to my question about computers. If these fibers can conduct electricity, act as logic gates, and assemble themselves, how do they integrate with computing?
This is where Enfield introduces the concept of the unified computational soup. Imagine a computer where DNA is the hard drive. We already established DNA is the densest storage medium possible.
Right, 200 petabytes a gram.
The problem is reading and writing to it quickly. Enfield outlines processing the DNA using terahertz radiation. Terahertz waves resonate exactly with the hydrogen bonds holding the DNA base pairs together, causing them to unzip and rezip in picoseconds.
So DNA is the storage, and terahertz radiation is the clock speed. But how do you wire millions of DNA strands together? Traditional silicon wiring is way too big and clunky.
That's the missing link. You use the self-assembling prion fibers. Because they are exactly 10 nanometers wide, they perfectly match the molecular scale of the DNA double helix. The prion fibers act as the physical logic gates and the wiring interconnects. The entire computing system operates at the sub-nanometer scale completely seamlessly without the inefficient, clunky interface between biological material and metal chips.
It's an entirely biological supercomputer. But I'm struggling to picture this from an engineering standpoint. There's a practical problem here. If prion amyloids are the perfect self-assembling building block, and they grow exponentially in all directions the moment they touch a seed, well, how do we tell them what shape to take?
That's the catch.
Yeah, if I just drop a seed in a vat of protein, I'm just going to get a big, chaotic clump of solid sludge. How does it become a computer circuit or an airplane wing?
You've hit on the exact bottleneck. This is the spatial control problem. Prion self-assembly is a bulk process. It can fill a volume, but it can't draw a line or write a pattern. To solve this, Enfield synthesizes a new paradigm called prion optogenetic lithography.
Let's break down those massive words. Optogenetic lithography. We're basically using light to tell the proteins where to assemble. We're writing matter with light.
Exactly. It rests on three interconnected pillars. First, the prion self-assembly provides the amplification, the physical material growth. Second, optogenetics provides the spatial control. And third, photolithography provides the precision patterning using lasers or holograms to draw the complex 3D shape we want.
Okay, let's dive into the optogenetics part because this is fascinating. Optogenetics is the use of light-responsive proteins that change their physical shape when exposed to specific colors of light. The text mentions engineering a chimeric protein using domains like LOV2 or Cry2. Let's not gloss over that. What is a chimeric protein, and how does a photon of light physically force a protein to change its shape?
A chimera in mythology is a creature made of different animal parts. A chimeric protein is a synthetic protein built by fusing two different functional parts together. In this case, scientists take a light-responsive domain like LOV2, which comes from oat plants, and they fuse it directly to our prion-forming domain.
And the light mechanism.
Inside the LOV2 domain is a tiny molecule called a flavin chromophore. It acts like an antenna. When a specific wavelength of blue light hits that antenna, it absorbs the photon's energy. That burst of energy causes a sudden chemical reaction. A covalent bond forms, which physically contorts a specific alpha helix in the protein, causing it to unfold.
It's like a molecular mousetrap. The photon is the mouse stepping on the trigger, and the protein instantly snaps open.
Exactly. In the dark, the LOV2 domain acts like a tightly closed lock. It holds the prion protein in a folded shape that cannot assemble. It just floats there as a harmless liquid soup of monomers.
Okay, but when the light hits it?
The instant the blue light hits it, the mousetrap springs. The LOV2 domain unlocks, the prion domain is released, it adopts its sticky cross-beta shape, and it instantly begins the templating cascade. The solid fibers grow, but only in the exact locations where the light touched the liquid.
The text mentions phobits, photoinducible binary interaction tools. These act as incredibly precise light-on and light-off switches. So you can not only start the crystallization with blue light, you can hit it with red light to stop it, or even dissolve it.
That's right. It gives you subdiffraction-limited molecular-level spatial resolution. Now, let's apply this. Go back to medicine. Imagine injecting a patient with a viral nanite that programs their cells to naturally produce these locked liquid chimeric monomers.
So their body makes the raw material.
Yes, the patient's tissue fills with the liquid precursor. Then, a surgeon inserts a microscopic endoscope and shines a highly complex holographic light pattern directly inside a tumor.
And the light instantly grows a permanent therapeutic scaffold exactly in the shape of the tumor. A scaffold that can release targeted drugs or use that piezoelectric effect we talked about to generate ROS and kill the cancer, while leaving the healthy tissue a millimeter away totally clear because it was in the dark.
Or consider neurology. You could use near-infrared light, which passes harmlessly deep into brain tissue, to write missing neural circuits directly into the white matter of a damaged brain. The holographic light draws the 3D pathway, the amyloid fibers crystallize instantaneously along that light path, and regenerating neurons use those fibers like a physical trellis to grow across a damaged area.
It's incredible. And this extends way beyond medicine into manufacturing and food. Enfield talks about protein lattice printing.
Yes. Think about the struggles of the alternative meat industry today. They can make plant protein taste vaguely like meat, but they struggle with texture. The snap of a shrimp or the chew of a steak comes from the exact microscopic architecture of muscle fibers, fat cells, and connective tissue.
With prion optogenetic lithography, you aren't just mashing soy protein together and extruding it. You have a vat of liquid protein, and you project a complex 3D hologram of light into it. Every voxel, every 3D pixel of the meat crystallizes exactly where the light dictates. You are building the microarchitecture of a steak with molecular precision, without an animal, and without even a mechanical print head.
And this is exactly what NASA is researching for deep space food replicators. You cannot physically carry enough prepackaged food for a multi-year Mars mission.
Right.
But if you carry dense, inert protein powder, water, and a light printer, you can print exactly the food architecture, the medical scaffolds, or the replacement tools you need on demand from the same feedstock.
So it's like 3D printing, but instead of a manticle nozzle clumsily squirting out hot plastic layer by layer over hours, you just flash a complex hologram into a pool of liquid, and the entire object crystallizes out of nothing all at once.
Exactly. It's instantaneous volumetric printing.
Okay, we've zoomed all the way down to the nanometer scale of hydrogen bonds and photons of light and built up to stakes and biological computers. Now, let's zoom all the way out to the continental scale.
The big picture.
Right. How does programmable biology save our collapsing planetary infrastructure? Because fixing a cell is one thing, fixing an ecosystem is another.
To understand the planetary application, we have to look at what Enfield calls the linear fallacy. This is the foundational categorical error of modern industrial civilization.
The idea that we treat biological cycles as linear transactions. We extract resources from the earth, we burn or consume them, and we discard the waste into a hole or a river.
Yes. And the text gives devastating, tangible examples of why this linear model is hitting a physical wall. Look at the Ogala Aquifer.
This one is terrifying. The Ogala lies beneath the Great Plains. It provides something like 30% of all the groundwater used for U.S. agriculture. It's the reason we can grow corn in dry environments.
But the Ogala is fossil water. It took between 6 and 12 million years of geologic time for rainfall and glacial melt to fill that subterranean sponge of gravel and sand. Because of our linear extraction, pumping it out millions of times faster than rain can replace it, up to 70% of the Texas Panhandle portion of the aquifer will be functionally unusable in 20 years.
Wow.
Parts of Kansas are literally facing what agricultural economists call day zero, where the wells simply suck mud.
We are mining water like it's coal.
Oh.
And then there's the nutrient cycle. We mine raw phosphorus out of the ground, and we use massive amounts of natural gas to synthesize artificial nitrogen fertilizer. We apply 16 million tons of it to fields in the Midwest, and because plants can't absorb it all, the runoff flushes straight into the Mississippi River system.
Which leads to the Gulf of Mexico dead zone. It's a textbook example of eutrophication. All that nitrogen and phosphorus hits the warm waters of the Gulf, causing an explosive, unnatural bloom of algae. The algae block out the sunlight.
And when they die?
When the algae inevitably die, they sink to the bottom, where billions of bacteria consume them. Those bacteria suck every molecule of dissolved oxygen out of the water, creating a 6,000 square mile zone where marine life literally suffocates.
We are extracting value on the farm, but externalizing all the catastrophic costs downriver. So how do we fix it? Enfield says we have to dismantle the linear system entirely and close the loops using biological infrastructure, starting with something called bio-CCS, bioenergy, with carbon capture and storage.
We currently treat industrial exhaust from a coal plant or an ethanol refinery as a waste problem to be vented into the atmosphere or expensively pumped underground. But exhaust is highly concentrated CO2. To a biological system that isn't waste, it's food.
Right.
So instead of a smokestack, you pipe that hot CO2-rich exhaust directly into open algae ponds or photobioreactors built right next to the power plant.
The algae literally ate the CO2, but how do they do it fast enough to keep up with a power plant?
Because microalgae are essentially the perfect biological machines for this. Unlike a tree or a soybean plant, algae are suspended in water. They don't need to waste energy or carbon building rigid stems, roots, or trunks. They're basically just naked chloroplasts dividing endlessly.
Highly efficient.
Extremely. Through photosynthesis, using an enzyme called rubisco, they strip the carbon out of the CO2 and use it to build their own cellular mass. They fix about 12 tons of CO2 per acre per year. They grow 100 to 200 times faster than terrestrial crops per unit of land.
What do we do with mountains of algae?
Half of their mass is lipids oils. You harvest the algae, press the lipids out, and that oil can be directly converted into drop-in biodiesel to run trucks and tractors.
So the coal plant's atmospheric waste becomes the carbon-neutral nutrient stream for a highly profitable local biofuel operation. Carbon isn't buried in a hole, it's cycled. But what about the water crisis and the fertilizer runoff? This is where Enfield introduces what might be my favorite part of the whole text: duckweed.
Duckweed is a phenomenal organism. It's those tiny, floating green leaves you see covering still ponds. It is the fastest growing vascular plant on Earth. Under optimal conditions, a mat of duckweed can double its entire biological mass every 16 to 48 hours.
That exponential growth curve is insane.
Hmm.
So how does it solve the runoff problem? If you channel agricultural runoff from heavily fertilized corn and sorghum fields into shallow, engineered duckweed trenches before it hits the river, what happens?
The duckweed acts like a biological vacuum cleaner. Its tiny rootlets hang down into the water and aggressively absorb the dissolved nutrients. It can remove up to 96% of the phosphorus and 99% of the nitrogen from the flowing water. It prevents the Gulf dead zone before it even starts.
And because it grows so fast, you have to constantly harvest it, right? You skim this massive explosion of duckweed biomass off the trenches. What do you do with it?
It depends on the water source. If you grew the duckweed in highly toxic wastewater or manure lagoons, you wouldn't eat it. You run that harvested biomass through an anaerobic digester, essentially a sealed vat with specialized bacteria, which breaks it down into methane biogas for electricity, and leaves behind a rich, clean biofertilizer that you put right back on the cornfields. The nitrogen loop is closed.
But the text makes a crucial point about separating streams. If you grow duckweed in relatively clean agricultural runoff or purpose-built ponds, it has a crude protein content of 35 to 45%. It rivals soybeans. It becomes an incredibly high-quality, ultra-cheap livestock feed for cattle or poultry.
Exactly. It's a dual-stream biological system that recovers the fertility that we currently just flush away into the ocean. And this brings us to Enfield's synthesis of the water problem, the concept of ditch 2.0, or hybrid irrigation.
This part truly blew my mind because it exposes how our obsession with mechanical efficiency actually destroyed the environment. Enfield explains the efficiency paradox of modern agriculture.
It's a tragic irony.
It is. Modern center pivot irrigation, those giant rotating sprinklers, are about 95% efficient at getting pumped water directly to the crop roots. They replace the old, inefficient, gravity-fed earthen ditches that farmers used for a century. But when farmers adopted precision sprinklers, it didn't save the aquifer.
No, the aquifer depletion actually accelerated because the farmers took the saved water and just used it to plant more acres of thirsty crops.
But it gets worse. Those old, supposedly inefficient, leaky dirt ditches were accidentally performing vital ecological services.
Yes, the water seeping out the bottom of thousands of miles of earthen canals was actually the primary way the aquifer recharged itself. And the standing water in those canals was constantly evaporating into the local atmosphere, humidifying the air, and providing the precise atmospheric moisture needed to trigger summer convective thunderstorms.
Unbelievable.
When we moved to concrete pipes and enclosed precision sprinklers, we inadvertently dried up the local atmosphere, killed the thunderstorms, and stopped the aquifer from recharging. We engineered the rain away.
So Ditch 2.0 brings the ditches back, but intentionally engineered this time.
Yes, you build shallow, terraced channels around the margins of agricultural fields, stocked with our friend duckweed. The duckweed treats the water and stops the nutrient runoff. The channels are designed with a massive surface area to deliberately evaporate moisture, priming the local climate for rain. And you engineer the bottom of the ditch with specific permeable gravel sections to deliberately and slowly recharge the aquifer. It's an integrated biological water treatment and evapotranspiration system, not just a dumb water pipe.
Let me stop and tie this all together because the genius of the text is how these layers stack. Where do we get the thousands of tons of cheap liquid protein feedstock we need to run those massive seeding vats that manufacture our amyloid structural panels, our biocomputers, and our light-written steaks?
You get it from the algae and duckweed that are actively scrubbing our coal plants and farm runoff. The whole stack is fully integrated. Layer 1 is the ecological infrastructure, duckweed and algae, providing the protein and energy while cleaning the planet. Layer 2 is the molecular delivery viral nanites programmed with CRISPR to act as the workers.
And layer 3.
Layer 3 is the hardware substrate prion amyloid materials tuned by hydrogen bond density. Layer 4 is the fabrication optogenetic lithography, using light to write those materials into usable shapes. And layer 5 is the resulting circular civilization.
Okay, I am sold on the science, but I have to ask the practical, cynical question. It sounds wonderfully utopian, it's beautifully engineered, but if it's so efficient, if duckweed cleans the water and gives you free fertilizer, why aren't farmers and utility companies already doing this? Why isn't there a duckweed trench on every massive farm in Iowa today?
Enfield points squarely at institutional inertia and misaligned incentives. First, look at the regulators. Environmental regulators are historically trained in civil engineering and chemistry. They prefer the mechanical certainty of a $50 million concrete water treatment plant with chemical dosing pumps.
Even if it's worse.
Exactly. They are deeply uncomfortable with the biological variability of a duckweed pond, even if it works better, because it's harder to model on a spreadsheet.
And the economics.
Agricultural subsidies currently reward maximum yield extraction at all costs, and they effectively penalize conservation. If you take an acre out of corn production to build a duckweed trench, you lose your subsidy for that acre. And fundamentally, the linear system hides its true costs. It is highly profitable to be a synthetic fertilizer company when you don't have to pay a dime for the dead zone in the Gulf of Mexico. If they were taxed for the ecological damage, biological circularity would become the cheapest option overnight.
Which brings us back to the ethical core of Enfield's vision. He calls it planetary citizenship. It's the idea that you don't just own a plot of land, you owe rent to the biosphere. The cost of the poison you produce must be paid in land restored. If you extract services from the earth, you are obligated by engineering necessity to maintain the biological systems that provide them.
It's an ethics of reciprocity, built directly into the engineering loop. Biology isn't a resource to be mined, it is a partner to be engaged.
So let's do a rapid-fire recap of the massive journey we've just been on. We started by reframing diseases not as invaders, but as corrupted code. And we stripped viruses down to their mechanical essence, incredibly efficient FedEx trucks waiting to be loaded with CRISPR repair manuals.
We moved into reading nature's evolutionary library, taking radiation resistance from tardigrades, oxygen efficiency from seals, or regeneration from salamanders, and using programmable evolution to instand those traits safely, bounded by strict ethical safeguards between somatic and germline edits.
Then we looked at the ultimate hardware building block, prion amyloid fibers. We saw how tuning their hydrogen bonds creates nanomaterials tougher than spider silk that can intrinsically conduct electrons through tyrosine hole hopping and generate power from sound.
Then we learned how to command those materials using prion optogenetic lithography, using holograms of light to trigger chimeric proteins to spring open and crystallize, printing everything from medical brain scaffolds to deep space food exactly where the light touches.
And finally, we zoomed out to the planetary scale, using algae to eat concentrated coal exhaust, using exponentially growing duckweed to clean agricultural runoff and produce free biofertilizer, and redesigning our irrigation ditches to literally bring back the rain and save ancient aquifers.
It is a profound, unified vision of a civilization that finally understands how to partner with biology at every level of scale, from the nanometer to the continent.
It really is. And I want to leave you with one final, slightly provocative thought that builds on everything we've talked about. We've focused today on top-tier scientists, FDA-approved gene therapies, and massive industrial agricultural loops. But if DNA is fundamentally just code, and light is just a compiler, well, what happens in 20 years when this biological technology becomes as accessible as learning Python?
That's a staggering thought.
Think about the history of computers. They started in massive government labs, and within a generation, they were in garages. What happens when a teenager in their garage isn't coding a video game, but is writing a light hologram sequence to grow a custom, self-repairing bicycle frame out of algae paste in a bathtub? The quill of life isn't just changing hands from blind nature to elite scientists. It is about to be open source to all of us. What will you write?
A fascinating, daunting question for an entirely new era of human history.
Thank you for joining us on this deep dive. I hope you walk away looking at the living world, the duckweed floating on a pond, the leaves on a tree, even the viruses we used to fear, not as a resource to be extracted or an enemy to be fought, but as an incredible masterpiece of engineering, just waiting for its next line of code. Because suddenly, that murky, unpredictable biological landscape we talked about at the beginning, it isn't a broken system anymore. It's a canvas. We'll see you next time.